Hey, how's it going? It's been a bit, huh? Hope the year was going good for ya. A few of you may have wondered where I went. Well, I got a job. Anyways, I realized that I kind of left you guys hanging, and a lot of you were messaging me asking me to make some more videos. Well, I didn't realize until we had about a week left until the AP test. So, this is the best I got. Hey, well, we're here, aren't we? So let's get to it. Here's how this is gonna work. We have a couple days left until the AP test. There's not much else for you to learn. So what I've decided to do is I've compiled every single topic that will be assessed on the AP test. You've probably learned a lot from your teachers this entire year. And so my goal is just going to be making sure you know exactly what you need to know for the test and what you might not necessarily need to have memorized. So, the objective of this video is to simply go over what you need to know and what you don't need to know. This will not be a video about teaching any specific topic. As a quick note, a lot of the things we talk about here you may have learned or you may have not learned. There may also be some things that you memorized and then realize now that you do not need to know them for the AP test. I'm not here to shade any of your teachers. They're doing what they do because they care. But every teacher teaches differently. They may be more comfortable at certain topics or less comfortable at other topics. So my goal in making this video is to just make sure that we know exactly what is going to be on that AP test. Another reminder, a lot of the AP Bio test is actually going to be application. So as long as you understand all the topics that I discuss in this video, you should then be able to take those topics and apply them to basically any scenario. Unit 1, the chemistry of life. Here's the vocab. And now we're on to 1.1, structure of water and hydrogen bonding. See this guy right here? This is the standard. As long as you can answer this question, then you should know everything that you need to know for this topic. So, water is composed of one highly electronegative oxygen and two hydrogens. This makes water a polar molecule, which means that it's more negative on one side and more positive on the other side. These positive and negative partial charges cause interactions between different water molecules. 1.2, elements of life. For this topic, you just need to know this chart. 1.3, introduction of macromolecules. You should know this chart. If you watched my Unit 1 video, you'll know that I kind of messed up with this chart last time. So make sure you use this one and not the one from the other video. The only other thing you need to know for 1.3 is dehydration synthesis and hydrolysis. The easiest way for me to remember is to think of hydrolysis. Hydro is water, lys to cut, and so you're using water to cut a molecule. Topic 1.4 to topic 1.7. All of these topics are pretty much the same, but each one is for a different macromolecule. You'll need to know the chemical composition of each, the structures and functions of each, and you should know the directionality for both the amino acids and the nucleotides. More or less, outside of polarity, this entire chapter is pretty much vocab. Unit 2, cells. By the way, you see this? This is roughly how many questions in this unit will be on the multiple choice, based on the topic distribution that's put out by AP Classroom. Here's the vocab, and now we're on topic 2.1, cell structure and function. For topic 2.1, you just need to know the basic structure and function of each of the cell organelles. Specifically, ribosomes, the endomembrane system, including the ER, Golgi, lysosomes, vacuoles, vesicles, nuclear membrane, and plasma membrane, mitochondria, and chloroplasts. Topic 2.2, cell size. Remember that when we're talking about cell size, we're talking about the ratio between surface area and volume. Higher surface area to volume ratio allows for better exchange of materials. If you keep the surface area the same, but you increase the volume, you end up with a lower surface area to volume ratio, and that will result in slower exchange. If you have higher surface area but keep the volume constant, you'll have a higher surface area to volume ratio, and that will result in faster exchange. This is why if you were reheating your burrito in the microwave, you could cut it in half to make it heat up faster. If you were wondering what all those surface area and volume equations were doing on your formula sheet, They are specifically for if you ever need to calculate the surface area to volume ratio in a problem. Topic 2.3, the plasma membrane. You should know the structure and function of a phospholipid. You should also know that because of the hydrophilic head and the hydrophobic tail, they'll come together to form a phospholipid bilayer. This pretty much results in a polar, nonpolar, and polar sandwich when in aqueous solution. And this phospholipid bilayer acts as a barrier for the majority of particles. You should also know what the fluid mosaic model is. Topic 2.4, membrane permeability. So, the plasma membrane prevents most things from diffusing through. You should know that small nonpolar molecules diffuse freely through the plasma membrane, and small polar molecules can diffuse, but in smaller amounts. Everything else has to move through either channel or transport proteins. You should also know that the cell wall provides structure. Topic 2.5, membrane transport. You should know what a concentration gradient is and that it's formed because of the selective permeability of a membrane. You should know the difference between passive and active transport, and you should also know the difference between endocytosis and exocytosis. A good way to remember is to think endo as into the cell and exo as exiting the cell. Topic 2.6, facilitated diffusion. As we mentioned, many particles can't pass through the membrane, but are extremely important for cellular activity. Channel proteins and transport proteins help move these particles through. Remember that facilitated diffusion means that there is no energy input. You should also know what aquaporins do. Topic 2.7, tonicity and osmoregulation. So this topic tends to trip up a lot of people, so I'm going to spend a little bit more time here. Remember that the terms hypotonic, hypertonic, and isotonic refer to the concentration of solutes in a solution. If something is described as hypertonic, that means the concentration of solutes is higher compared to the hypotonic solution that has less solutes. So what this means is that these terms are relative, kind of like if somebody is taller or shorter. Typically in a question, you're going to see hypertonic or hypotonic referred to the environment surrounding a cell. If the environment outside the cell is hypotonic, that means that inside the cell it's going to be hypertonic. If the membrane prevents solutes from moving through, then water will always move from hypotonic solutions to hypertonic solutions. You probably also did some water potential equations when you went over this topic. Most of the questions that you'll see involving this topic will likely do with the concept, but the equations are fair game and you should know how to solve problems using them. Topic 2.8, mechanisms of transport. If you're moving something against its concentration gradient, that's going to require energy, and that is known as active transport. You should be familiar with the sodium-potassium pumps and ATPase. Topic 2.9, cell compartmentalization. You should know that eukaryotes have membrane-bound organelles that help promote certain metabolic processes and increase surface area. Topic 2.10, origins of cell compartmentalization. You should know the endosymbiotic theory for mitochondria and chloroplasts. You should also know that prokaryotes don't have membrane-bound organelles, and instead just have certain regions that they do specific processes. Unit 3, cellular energetics. Here's the vocab. Topic 3.1, enzymes. You should know that enzymes are catalysts, and you should know that they lower the activation energy. You should know that making or breaking bonds requires a little bit of energy to start, and that's known as the activation energy. You should know what an active site is, and that it has various properties that allow it to specifically interact with certain substrates. Topic 3.2, environmental impacts on enzyme function. You should know that proteins are highly dictated by their shape. You should also know that enzymes are proteins. You should know that there are different types of environmental changes that can actually denature an enzyme and prevent it from working. You should also know that there are some environmental changes that can slow down or speed up the enzyme's efficiency. Topic 3.3, cellular energy. For this topic, you just need to know that cellular processes are always releasing energy. This is why we need a constant input of energy in order to survive. In order to do this, we use metabolic pathways, which efficiently transfers energy through different processes. The most basic metabolic pathways are actually seen throughout every domain of life. If you learned anything about Gibbs free energy, that will not specifically be assessed on the AP Bio test. 3.4, photosynthesis. So for photosynthesis, here's what you should know. The photosynthesis equation, the general structure of chloroplasts, You should know the leading theories of how chloroplasts evolved. You should know the general steps of the light-dependent reactions, and this includes the inputs and the outputs. Specifically, you should also be familiar with the electron transport chain and how the proton gradient works there. You should also know where each reaction within photosynthesis occurs within the chloroplast. For things you don't need to know, you do not need to memorize all the names of the enzymes or the intermediates. Additionally, you do not need to memorize every single step of the Calvin cycle. The only thing you need to know is the inputs and the outputs. Alright, topic 3.5, cellular respiration. You should know cellular respiration, respiration versus fermentation, the electron transport chain, and the proton gradient as it pertains to the mitochondria. You should be familiar with decoupling oxidative phosphorylation. And for glycolysis and the Krebs cycle, you only need to know the inputs and the outputs. Again, you do not need to memorize all the names of the enzymes or the intermediates. You also do not need to memorize all the steps of glycolysis or the Krebs cycle. For the most part, everything in this picture you should look at and be familiar with. Unit 4, cell communication and cell cycle. Here's the vocab. Topic 4.1, cell communication. You should know that cells talk to each other through chemical signaling. You should know the difference between a short distance signal and a long distance signal. And also bear in mind that any examples that you went over with short distance or long distance signals do not need to be memorized. However, you don't know what kind of application question AP Classroom is going to throw at you, and so knowing more examples can always help. But if you don't know any examples, you should be able to take the concepts and apply them. Topic 4.2 and 4.3, signal transduction. In general, you should know signal, transduction, response. Every single example that you learned about in class will follow these 3 steps. Some specific examples that you should be familiar with are the G protein coupled receptors, and you should also be familiar with cyclic AMP. If you see a question on this topic, the response will typically be given to you. It'll be your job to predict how changing a specific part of the cascade will affect the response. Topic 4.4, feedback. For topic 4.4, you should know that negative feedback promotes stability and positive feedback promotes transformation. Both of these are necessary in order to conduct regular cell activity. Topic 4.5 and 4.6, cell cycle and its regulation. You should know all the steps and checkpoints of the cell cycle. You should also know all the steps of mitosis. You should be familiar with how cyclin and CDK control the cell cycle, but you don't need to know any specific pairs. Unit 5, heredity. Here's the vocab, and we're off to topic 5.1 and 5.2, meiosis and genetic diversity. For meiosis, make sure you are comfortable with the vocabulary. There's a lot of vocabulary here and they're all intertwined with one another. A lot of my students get this one mixed up, so make sure you're comfortable with it. You should know the steps of meiosis and you should know which parts of it promote genetic variation. You should be familiar with non-disjunction and you should know what can happen if it occurs during different parts of meiosis. You should also know the difference between mitosis and meiosis. Topic 5.3, Mendelian genetics. This one also has a lot of vocabulary, but a lot of it you might have seen before. You should know the difference between gene versus allele, genotype versus phenotype. You should know unlinked genes versus linked genes. You should also be familiar with Mendel's laws. And you should also know what a test cross is. In terms of Punnett squares, there will not be a specific question where they ask you to complete a Punnett square. Instead, most of the questions you'll see involving Punnett squares will involve the application. For example, they will show you some sort of scenario and you'll have to use the Punnett square in order to make a prediction. When you went over this topic, you probably also covered dihybrid crosses. Dihybrids can get super confusing, and because we don't do it often, it's better to use the probability rules than to draw out the 16 squares. So for example, let's say you had a plant. It could be tall or short, or leafy or not leafy. And the scenario asked you to do a dihybrid cross between 2 plants. Instead of drawing out all 16 squares, you'd make one Punnett square using only the tall or short phenotypes, and you'd do another Punnett square for the leafy or non-leafy phenotypes. Then you would use the laws of probability to combine those two outcomes. So if you wanted the probability of a short and leafy plant, you take the probability that you got for the short, and then you'll take the probability that you got for leafy, and then you'd multiply them together. If you needed the probability of short or leafy, then you would take both of those probabilities and add them together. Topic 5.4, non-Mendelian genetics and environmental effects on phenotype. You should know the difference between incomplete dominance and codominance. Something small that helps me remember is ink pink. If you had red and white flowers, incomplete dominance would result in pink flowers. You should also be familiar with sex-linked traits, linked genes, non-nuclear inheritance, so that's the mitochondrial or chloroplast, and you should also be familiar with pleiotropy. Make sure you know how to read a pedigree, and you should also be able to look at a pedigree and know what type of inheritance it is based on what you see. You should also be familiar with the term phenotypic plasticity. Unit 6, Gene Expression and Regulation. Here's the vocab for the unit. Topic 6.1, DNA and RNA structure. A lot of this is reviewed from previous units, but you should be familiar with the difference between DNA and RNA. You should know how DNA works in prokaryotes versus eukaryotes. You should be comfortable with the base pairing rules, and you should also know how directionality works in both DNA and RNA. Topic 6.2, DNA replication. DNA replication occurs in the S phase. The only enzymes that you need to be familiar with are these right here. You should also know that DNA is replicated in a 5' to 3' direction, and you should be familiar with the term semi-conservative. Topic 6.3, transcription and RNA processing. Be familiar with the central dogma, DNA to RNA to proteins. You should also know the difference between mRNA, tRNA, and rRNA. You should be familiar with the difference between the template strand and the coding strand, and you should know what a poly A tail, a GTP cap, introns, and exons are. Topic 6.4, translation. You should know that ribosomes are responsible for translation, that in prokaryotes, translation and transcription can occur at the same time. You should be familiar with the steps of translation, and again, you do not need to know the names of any specific enzymes or codons. The only codon that you need to be familiar with is AUG, which is the start codon. You should also know what a retrovirus is. Topic 6.5 and 6.6, gene expression. You should know that some genes are necessary for cell maintenance and are on all the time, while other genes can be toggled on and off. You should be familiar with the term regulatory sequences, and you should know that epigenetic changes affect expression by modifying the DNA or through the use of histones. You should know what transcription factors are, and you should be familiar with both the lac and trip operons. And you should know that different transcription factors will allow the expression of different DNA, which will result in different cells. Topic 6.7, mutations. The mutations that you should be familiar with are point mutation, frameshift mutation, silent mutation, and nonsense mutation. You do not need to know specific mutations and their effects. For example,
the mutation of the BRCA1 gene will cause blah 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 blah.
Remember that what makes a mutation good or bad entirely depends on the environmental context. You should also know that mitotic or meiotic errors can also cause changes in phenotype. You should know how the DNA can be changed in prokaryotes, viruses, and in the sexual reproduction process in order to promote genetic variation. Topic 6.8, biotechnology. You'll need to be familiar with gel electrophoresis, PCR, bacterial transformations, and DNA sequencing. That one's the one where you compare those bars and try to figure out which one's the father.
Collins, you are not...
You should understand when and why you would conduct each and every single one of these processes. However, you do not need to know any specific steps on how these processes are done. It's good to know, especially if you're planning on pursuing a career in biology, but it is not going to be assessed on the AP test. Unit 7, Natural Selection. Here's the vocab. Topic 7.1, 7.2, 7.3, and 7.11. Natural selection, and I have no idea what I'm doing anymore. You should be familiar with Darwin's theory of natural selection. As the environment changes, this affects evolution through selective pressure. You should be familiar with the term fitness, and that certain phenotypes can increase or decrease fitness. And you should also know that the variation in the number and types of molecules within cells can also increase the chance of survival. As for artificial selection, you should know that humans can also affect evolution, specifically by making other things do whatever the heck we want. Okay, here are some common misconceptions, so it's really important to remember this. A single individual of a species cannot evolve. Additionally, species don't intentionally mutate. Through variation within populations in a species, the species as a whole can evolve over time as certain phenotypes die out and certain phenotypes reproduce more. Topic 7.4, Population Genetics. You should be familiar with genetic drift, the bottleneck effect, the founder effect, and migration. You should also know how all of these things affect the genetic makeup of a population. You should know that mutations, genetic drift, and gene flow can all affect allele frequencies within a population. This change in allele frequency is proof of evolution within a population. Topic 7.5, Hardy-Weinberg equilibrium. So, you should know the conditions of Hardy-Weinberg equilibrium. You should also know why Hardy-Weinberg equilibrium is even important. It provides us with a null hypothesis to compare allele frequencies when we do testing. Now remember that allele frequencies are going to be P and Q. The genotype frequencies are going to be the P squared, 2PQ, and Q squared. Most of the time, these questions will give you P squared, 2PQ, or Q squared. Your job is to then find either P or Q. The easiest thing to do is to find Q squared. From there, you can square root it to find Q, and then you can find P. Remember that Q squared is the recessive phenotype. Topic 7.6, 7.7, and 7.8, Evolution. So these topics basically just go over the many different ways that we have proven that evolution is real. You should be familiar with homologous versus analogous traits, and you should also be familiar with radioactive dating. Topic 7.9, Phylogeny. You should know the difference between a phylogenetic tree and a cladogram. You should be able to look at either one and be able to determine evolutionary relatedness. You should also know how to make a cladogram based off of a data table. If you haven't done that before, I would try it just one time to make sure you understand how to do it. Topic 7.10, Speciation. Speciation occurs when two populations become reproductively isolated from one another. You should be familiar with divergent versus convergent evolution, sympatric versus allopatric speciation, and prezygotic versus postzygotic barriers. Topic 7.12, Origins of Life on Earth. For this topic, you need to know how old the Earth is and how old the earliest fossil evidence of life we have is. You should also be familiar with the RNA world hypothesis. Unit 8, Ecology. Here's the vocab. Topic 8.1, Responses to the environment. For this topic, you probably saw many different examples. Just know that you are not required to memorize or know any specific example that you saw. All you should know is that organisms have a bunch of different ways to communicate with the world around them. If an organism's behavior promotes survival or reproductive success, then those traits will be passed on. Topic 8.2, Energy flow through ecosystems. You should know the difference between an endotherm and an ectotherm. And you should know autotrophs versus heterotrophs. Now, the kind of annoying thing about topic 8.2 is that you should be familiar with all of the chemical cycles that are in this topic. All of these are fair game. You do not need to memorize the steps in any of these cycles, but you should know that energy and matter flows through each of these cycles, and you should know the general idea of what each cycle is about. Topic 8.3 and 8.4. Population ecology and the effect of density on populations. So for the next upcoming topics, there are a couple equations that you're going to run into. Each of these equations you should be familiar with using. From what I've seen of the AP test, if you do get a question using one of these equations, they're not going to be too complicated. So don't worry too much about mastering these equations. You just need to be familiar with the variables and knowing what goes where. Now, for topic 8.3 and 8.4, you should know that birth rate, death rate, and the population size can all affect the growth of a population. You should also be familiar with density-dependent and density-independent factors. Topic 8.5 and 8.6, community ecology and biodiversity. You should know the difference between communities and populations. You should know what predator-prey interactions are. You should be familiar with cooperation, trophic cascades, and niche partitioning. Be familiar with the terms competition, predation, mutualism, parasitism, and commensalism. You should know what a keystone species is and how it affects the environment. And you should know that the more components there are, or the higher diversity there is in an environment, the more resilient it will be. Topic 8.7, disruptions in the ecosystem. You should be familiar with the term heterozygote advantage. You should also know how an invasive species can affect an environment. You should also know how humans can impact the environment. You should be familiar with the terms biomagnification and eutrophication. And you should know the main geological and meteorological is that even a word? And you should be familiar with the main geological and meteorological events. And that's all of AP Biology. Not that bad, right? But on a serious note, if there's anything that I mentioned that you might not have seen before, it might be good to just brush up on it a bit. Now, before I end this video, I kind of wanted to leave you with a little bit of advice. For some of you, this might be your first AP test. For others, you might think that if you do bad on this test, you will never fulfill your dream of becoming a doctor. And I'm here to tell you that you have nothing to worry about. Don't get me wrong, AP tests are really important. And it's a little corny, but you're not gonna really care about the score you got on your AP Bio test 10 years from now. Also, how well you do on this single test is not a determining factor of how successful your life is gonna be forever. If you're watching this video, you're probably feeling a lot of pressure for that test right now. Because if you're watching this video, oh man, you must be desperate. But also, if you're watching this video, that means that you care a lot about your future, and that you probably have a good head on your shoulders. The best advice that I ever received when I was in high school was that you're gonna be fine. As long as you're out there trying your best, doing the best you can to be successful, you will eventually find a way to be wherever you need to be. And so I hope that no matter how you're feeling right now, you can take a deep breath and just tell yourself that you're gonna be okay. Alright, and that's all I've got. Best of luck on the AP test. Sorry for disappearing on all of you guys. And